Hello, everyone, and welcome to the first of our online lectures. As I mentioned on our Moodle, I'm going to be pre-recording our lectures so that we can use our class time for questions and discussion as required. You're also welcome to watch these lectures during class time and hop into either an earlier or later Google meeting if that's better for you. But the goal here is to give you a combination of flexibility with multiple options for you to choose from without losing the structure that comes from meeting in person. Because I'm still going to be here Monday, Wednesday, and Friday in Google Meet to answer questions. So hopefully we find a good compromise, at least for the next little while we're online. But these lectures should be pretty much the same as what you would get in class, just minus my fabulous hand gestures and probably plus a few bird background sounds. What we're going to do now is start looking at some of the other parts or the other things that we haven't actually addressed yet. So some diagrams will show what's called an axon hillock, and this is basically the point where our axon joins with the cell body or the soma. So some diagrams will indicate that for us. It matters a lot more when you get into some of the nitty-gritty about how cells fire. For us, we're just going to care that that's the part where the axon connects to the cell body. The next thing that we can point out is the myelin sheath. So myelin is a fatty, whitish insulation layer that's derived from glial cells during development. So this is going to wrap around our axon and basically act as insulation. So if you think of the plastic coating on an electrical wire, how it prevents the electricity from seeping out, these pieces of myelin sheath, this myelin, isolates the axon and all of the stuff that's going on inside it from the rest of the environment. It also does something really cool because we have these gaps between each of these sort of bundles of myelin sheath, and these gaps are called nodes of Ranvier. And so that's a place where the myelin sheath is very thin or even absent. And where it's open like that, that's where the cell's surface, the cell membrane, is exposed to the environment. So what happens is because myelin is sort of tightly insulating the axon in between these points, we can't have action potentials generated underneath the myelin. So the cell doesn't even have, say, protein channels or ion pumps under there. They don't need them because with that insulation, it allows those charges, it allows the signal to transmit and jump to the next gap. So at that next node of Ranvier, we have a gap, we have exposed cell membrane, and at that point, we have ion channels that can then be opened and pumps that can reestablish that balance. So we have action potentials sort of jumping from node to node to node. So it would go from here, and then there's no sort of new action potentials until we get here, and then we have new action potentials generated, and then we hop ahead, and then the new ones go here, and so on and so forth. And this is actually a really efficient way of doing it. First off, because it's going to save time, because we don't have to go through the process of an action potential firing at each and every single point along the whole length of our axon. Instead, they only have to fire at the points where there isn't myelin. So it can fire here and then skip over all of this stuff and fire here and skip and fire. So we save lots of time. And that actually allows signals in our neurons to fire much faster than they would otherwise. This also saves us energy. Because as you can imagine, if we are pumping ions sort of in and out of the cell and setting up these gradients and then having all of that function, that would be using up a lot of energy in the cell. So if we don't have to generate action potentials anywhere that the myelin is covering, then we get to save that energy as well. So the myelin sheath, this extra insulation, lets us move faster, it lets us save energy, and it lets us get our signals where they need to go a lot quicker without interfering with other parts of the cell. The next thing that I want to mention very briefly is glial cells. We don't get into a ton of details on glial cells other than the fact that they sort of put the myelin sheath in place. They're also there to help support and surround our neurons. So they guide neurons into the right places as they develop. They hold them in the right place. They get rid of debris. They bring in nutrients. So glial cells are a fantastic support network for our neurons. But we don't get too much into them because we could spend a lot of time talking about all the different kinds of glial cells. So I'm just acknowledging them here so that we can move on. Okay, so we've spent all this time talking about how action potentials occur and what's going on within a cell, but we've only hand-waved the fact that this communication happens between cells. So how does the action potential's information get from one cell to the next? So first, I'm going to introduce a little bit of terminology here. So when we have, actually, let me grab a pen here so I can draw, because I find that a little bit easier. So if we have our neuron, and it has the axon and the terminals at the end, and then it's probably got some dendrites over here. So the signal is always going to be moving in this direction. It's always going from the dendrites to the axon. And so once we get to the terminals, the axon terminals, at the very, very bottom, we have what are called terminal buttons, which are basically just these little protuberances. They look like little balls on the end of the axons, and that's just to increase surface area to help us transmit the signal better to the next cell. So the next cell is going to be another neuron. It's going to have its dendrites lined up sort of over here, and then it'll have its axon and then its own terminals down there. And so we're going to be looking at this part in the middle where we jump from one cell to the next. But I just wanted, as I was saying, to introduce some terminology. So this gap is called a synapse. So the terminal buttons of this axon don't actually touch the dendrites of the next neuron. They come very, very close together. There's a small gap here, and that gap is called the synapse or the synaptic cleft. So it's just a fluid-filled space that exists between those neurons. If we wanted to talk about the neuron that comes ahead of the synapse, we can call this the presynaptic neuron, because it's the one that's in front of our synapse as the signal flows. This second neuron, the one whose dendrites are at the synapse, that would be our postsynaptic neuron. And that's because it's the neuron that follows the synapse as that signal travels. So if we talk about a presynaptic neuron and a postsynaptic neuron, we're just talking about the order they come in this signal process. So when we're looking at this diagram here and we have the axon and specifically the terminal button of that axon here, that's the axon and the terminal button of the presynaptic neuron. Then we have the synapse, which is the gap between the two neurons. And then we have the dendrite, these parts of the postsynaptic neuron. So the neuron that's after our gap. And so our signal is transmitting in this sort of a direction, but this is where we're going to start talking about how do we go from an electrical signal, which was our chemical charges in the axon, and then we're going to actually look at turning it into a chemical signal to go between the cells, and then we're going to turn it back into an electrical signal once we're in the next neuron. So it goes electrical, then chemical, then electrical. And we've already talked about the electrical part because that's our action potentials. So now we're going to zero in and focus on this chemical part and start talking about neurotransmitters, which are the chemicals that carry the message from our presynaptic neuron to the postsynaptic neuron. And then just one more thing I want to point out while we're looking at this image here, we have these blue circles. These are called vesicles, and they're basically sort of bubbles or sacs that contain our neurotransmitters. So they have lipid membranes the same as the cell does. And then what they can do is if they come up to the cell membrane, they can bind with it and open up to dump their contents from inside the vacuole. They'll then dump it outside into the synapse. So that's the basics of what we're going to talk about here. All right, so let me get rid of our visuals there, and then we can hop to the next slide. So when we're talking about our neurotransmitters, I already kind of gave the definition, but they're chemical substances. They're going to carry messages across the synapse, and they can serve one of two functions. They can either excite the next neuron, or they can inhibit the next neuron. So exciting, or being an excitatory neurotransmitter, means that you make it more likely that that next neuron fires. If you inhibit firing or are inhibitory, you make it harder for the next neuron to fire. So if we think of our sort of local potentials, if we're going to be more likely to fire, then we are going to increase, sort of repolarize that membrane. Sorry, I said I mean depolarize. Let me hop to the image that I'm thinking of. So with our excitatory neurotransmitters, and I'm going to grab my visuals because I find these help. So we have our sort of cell at its resting potential. So this is our postsynaptic cell. And so these neurotransmitters are what are creating those local potentials. So if we're making it more likely for the next cell to fire, we are making this cell more positive. We're getting it closer to that 0 mark because it starts at negative something, right? So when we're depolarizing this membrane, we're causing that increase in charge, hopefully enough to hit our threshold. And if we get enough of these excitatory neurotransmitters, if they cause large enough local potentials, they're going to trigger our action potential. So it increases the likelihood of an action potential because it's reducing the polarization or making the cell membrane more positive. If instead, get rid of that, if instead we're looking at inhibitory neurotransmitters, can change colors to make it a little easier, they're going to be making the cell more negative. So we can talk about it hyperpolarizing the membrane. So the local potentials, instead of getting higher, are making the charge even lower. So in order for this cell that's been inhibited to fire, we would have to get a ton of excitatory signals to bring it back up just to hit the threshold before we could fire. So by making it more negative, by hyperpolarizing that membrane, we make it less likely that an action potential is going to occur. We're making the cell more negative and making it harder for that cell to fire. So just another way of saying it, excitatory neurotransmitters increase the probability of a neuron becoming active or firing, and inhibitory neurotransmitters decrease the probability of a neuron becoming active. And these are just a couple of examples of some well-known excitatory and inhibitory neurotransmitters. I will point out that dopamine can do both, which is just lovely and confusing. But depending on what kind of site these neurotransmitters bind to, they can create different effects. That's one of the things that allow us to have such complex signals in the brain, is that we have different neurotransmitters that can be released from different places, but we also have different receptor sites that they can bind to, and when they bind, they can create different effects, which is how dopamine, if it binds to a receptor that creates an excitatory situation, it can cause that increase in the charge so it can get closer to 0. Or if it binds to an inhibitory receptor, it can sort of cause it to be more polarized. It can go in the opposite direction. And just to talk about how our neurotransmitters might bind to receptors, we have another visual here. And again, I'm sort of pulling things from multiple different books because I like different visuals to illustrate different points. So this does a great idea of showing us this presynaptic neuron. They're also calling it a sending neuron, which might help you visualize. And our postsynaptic neuron, the one that's receiving a message. And then we're going to blow up this synaptic space, our synapse, where we have our presynaptic axon terminal, or specifically the little bulb on the end being those axon buttons or terminal buttons. And then we have the postsynaptic membrane, so this would be the dendrites of the next neuron. And so we have our vesicles. They're going to move sort of towards the end, and when they're triggered, they can bind to that cell membrane and dump the neurotransmitters out into this synaptic space. We're going to talk on the next slide about the receptors, but just the basics of it is that different neurotransmitters are going to be different shaped molecules. So they've simplified it here with balls with different sized and shaped sort of pointy bits on them. That's kind of the same idea. These molecules will all have different shapes, and they'll be able to bind to different receptors. So if this neurotransmitter fits this receptor, it can bind and activate that receptor, triggering whatever the effect is on the other side. So maybe it's going to open up some sodium ion channels and cause an increase in the charge. It's going to depolarize that membrane. But if the neurotransmitter doesn't fit the receptor, then it can't bind there, and then it doesn't activate it. And if we want to look at the process of sort of setting this thing up, you usually start with the synthesizing your neurotransmitters. So the cell will be set up to assemble them, put together all the moving parts, and then they store them in these synaptic vesicles. So these little sort of bubbles that contain those neurotransmitters. The next step is to release them out into that synaptic space, out into the synapse. And then if they find receptor molecules they can bind to, they'll bind to those receptor sites. And then once they've bound, there's usually a final step that involves cleaning up. So you don't want these receptors always on, so you want to deactivate them. So you can either re-uptake your neurotransmitters. So there are special systems in place where things will go around and collect the neurotransmitters and put them back into vesicles to be reused, or they might break them down. So you might see an enzyme that comes along and maybe cuts up all of the neurotransmitters so that they can be recycled and turned into something else. But in either case, those deactivate or disconnect our neurotransmitters so that they only act in a very specific area for a very specific amount of time. And just another example of what we're thinking of with our neurotransmitters binding to a receptor. This is from our textbook, and it shows that the neurotransmitter can sort of fit into a slot, kind of like a lock and key. So the key is like the neurotransmitter. If it has the right shape, it will activate or bind at that receptor site and be able to turn it on or open the door. So that's kind of how we think about it. And then very, very briefly, I just want to talk about how drugs can start imitating or acting like neurotransmitters, or at the very least, affecting how our neurotransmitters function. And they tend to do this by activating the same receptors that our neurotransmitters do. So drugs can be created that either replicate or prevent the action of our receptors. So if this drug is what's called an agonist, actually I have it on the next slide, so we're going to talk about the terms sort of interchangeably between the two. So if the drug is an agonist, then it's going to replicate the receptor action. So what that means is when it binds to that receptor, it's going to behave as though the neurotransmitter that belongs there has bound, and it's going to do whatever it was supposed to do. So if this is supposed to be a dopamine receptor, and we have a drug that comes in and binds as if it was dopamine, and this site opens or starts activating as though it was bound by dopamine, then we can call that an agonist. We can also have what's called an antagonist, and that's when the drug prevents the receptor from acting. So what happens there is that the drug would bind and sort of prevent dopamine from doing anything. And it can work in two ways. It can either be direct or indirect. So I've been kind of pointing at our competitive or direct competition. So that's when the drug binds to the neurotransmitter's binding site and either activates it if it's a direct agonist, or it binds to that site and turns off this receptor. That would be a direct antagonist. So we could even draw in the fact that it turns it off. It doesn't act the way that it should. And then if it's an indirect competition, then we have a neurotransmitter site, but the drug can bind elsewhere. So if it's an indirect competition or non-competitive binding, then it's not going into the same site. It's going to bind somewhere else on that molecule. But if it's an indirect agonist, if it binds to that other location, it can still turn on our site. It can turn on our receptor. And if it's an indirect antagonist, then it's going to bind somewhere else and it's going to turn off or prevent this neurotransmitter from making this active. So that's how drugs can either sort of imitate and act as though they are our normal neurotransmitters, or they can act in a slightly different way and just cause similar effects by activating or deactivating the same receptors. And again, you can take entire courses on drugs and neuroanatomy and stuff like that, but we just keep it very, very brief for this course. So we can leave it there for this topic. And then next we're going to start talking about how we organize all of these neurons together to create our nervous system. Actually, before I move on to the nervous system, I did have a few terms that I wanted to mention because they appear in our textbook. And you're going to see that these overlap quite a bit with stuff we've already talked about, just with slightly different names. So when I talked about a neurotransmitter, or a drug, I suppose, binding to a receptor and causing a change to the cell that receptor's in, i.e. excitation or inhibition, we can then call that change a postsynaptic potential, because it is a change in charge or voltage that's happening at a receptor site on the postsynaptic neuron. So that's what it's saying here. So these are actually the graded local potentials that we were talking about earlier on. So they can build up and cause an action potential, or they can they can make an action potential less likely to occur. So basically, they're going to change the probability of that postsynaptic neuron firing. And they're not all or none because because they're graded. They can build up, they can add together, and they can cancel each other out. So if you get a positive postsynaptic potential, positive PSP or excitatory PSP, that's causing an increase in the voltage, so that's getting us up closer to zero, and that's going to make the action potential more likely to occur. We can also have a negative voltage shift where we get more negative, and that would be inhibitory. It makes that action potential less likely to occur. And if you have some positive and some negative, then they can equal each other out. So they'll add together and might not net any difference if there's enough positives and negatives to cancel each other out. But just those are some terms that the textbook uses that I realized I didn't include earlier on. Another thing that I very briefly wanted to mention is this idea that a single neuron, even though we've been talking about them as sort of a presynaptic neuron and a postsynaptic neuron and nothing else, sort of a straight A leads to B, it's not actually that simple. One neuron can receive signals from thousands of other neurons across thousands of different synapses that connect to all sorts of different places on the cell. So each neuron can then integrate all of those signals that are arriving at the same time or in the same place before it makes that decision to fire, meaning that it's reached that threshold to fire. So even though we talk about everything in that simplified sort of one neuron leads to one other neuron, it's a lot more complex than that. So you might see talk of neural networks, where we start looking at patterns of neural activity, which means patterns of neurons firing, sort of as they connect to one another in very complicated fashion. So there's this interconnected web of neurons that are going to fire either together, meaning they fire at the same time, or they fire in a particular pattern. And those patterns seem to be linked to sort of what we're thinking or how we're thinking. That's where there's a pretty big disconnect between our understanding of how things work at the cellular level and how we turn that into actual meaning at the level of consciousness. But just sort of acknowledging that that is a thing that we should also be aware of. And then our last point to point out is the fact that these networks are constantly changing because our synaptic connections aren't completely stable. So we have what's called synaptic pruning, which is basically where we can eliminate old or unused synapses, or we can create new synapses, new connections between neurons if they start firing more frequently together. So it's not a static, solid network that's present from birth. What we actually see is a pretty dramatic change in the number of synaptic connections in the brain as we age. And there's a neat little graph here that shows, this is just looking at the number of synapses in the human visual cortex, starting from sort of pre-birth, and then as soon as we're born, we form tons and tons and tons of synapses, and this is massive. This is times 10 to the power of 11, so this is many, many, many synapses. So we build up tons of them. We kind of peak around like 8 months old, and then we start pruning. So that's the process of synaptic pruning, where we only keep the ones that are useful, and we start setting up those patterns, those networks that we need to continue for the rest of our lives. In the next section, we're going to start talking about the organization of the nervous system. And to ease our way into this fairly large topic, we're going to start talking about the peripheral nervous system. And so I suppose step one is to talk about what is the peripheral nervous system. So when we look at our nervous system, we divide it up into two broad categories, the peripheral nervous system and the central nervous system. So the central nervous system, which we'll talk about later on because we go into that part of the nervous system in a lot more detail, refers to the brain and the spinal cord. So even though that's only two things to talk about, we are going to talk about all the different areas of the brain and the really important parts of the brain. So that's going to be a more detailed lecture to come. But we're going to start, as I said, with our peripheral nervous system, which is basically everything other than the brain and spinal cord. For those who want to keep track of the topics that we're going to discuss, this diagram here does a pretty good job of showing our division of the nervous system. So we have our central nervous system and our peripheral nervous system. So once we get into the central nervous system, we're going to talk about all the different parts of the brain, sort of broken down like this. We'll also talk about the spinal cord. But as I said, we're focusing on the peripheral nervous system in this lecture. So we're going to focus in on all these divisions here. We're going to talk about the somatic versus autonomic nervous system. And then within each of those, we're going to break them down into all of those components. So this is just an overview. It might not make any sense right now, but as we work through all of this, this image is going to come back up multiple times as we work our way down this entire pattern here. So let's start, as I said, just with our peripheral nervous system and then the divisions of that. So as I said, the peripheral nervous system are the nerves that lie outside of our central nervous system, everything that isn't the brain and the spinal cord. Our two major divisions of the peripheral nervous system are the somatic nervous system and the autonomic nervous system. So our somatic nervous system is what's responsible for all of our voluntary muscle control and our sensory receptors. So when I say voluntary, I mean like when we intend to move. So if you think about clapping your hands, that would involve your somatic nervous system. In contrast, our autonomic nervous system, sometimes shortened to ANS, controls our automatic or involuntary functions. So I always remember this as the autonomic system is automatic. So we don't think about doing these actions, it's just going to happen. So this would be things like breathing, our heart rate, blood pressure, things that happen internally without us being consciously aware of it. That's all controlled by the autonomic nervous system. And if we hop back to our diagram here, so we've got our somatic and autonomic nervous system, we can also divide our autonomic nervous system into sympathetic and parasympathetic branches. So our sympathetic part of the autonomic nervous system, so it's happening without our control, is sort of our go system. That's where we have our fight or flight responses. So when we are autonomically aroused, this would be sort of a speed up, an increase in heart rate, an increase in metabolism, an increase in breathing rate. All of those things are related to the sympathetic nervous system. And then parasympathetic is the opposite. So that's our stop system. So once we're out of a situation that activated our sympathetic system, once we are ready to calm down and relax, we'll activate our parasympathetic system to calm our bodies back down again. So this would slow our heart rate back to normal. It would reduce our blood pressure. It would help our breathing get back to normal. So these two act kind of in combination and in opposition of one another to keep us at a normal balance. For those who are more visual folks, we have this image here that shows the parasympathetic versus the sympathetic parts of the nervous system. So it shows that we'll start with the sympathetic system because that's what we talked about first. So we're in fight or flight mode. Our pupils will dilate, which just means that our pupils will open wider, which will allow more light in, which allows us to see more detail, which is important if we're, say, being faced by a predator. We want to be aware of our environment. And for the contrast, our parasympathetic would then constrict the pupil, bring it back to normal. Our sympathetic system will inhibit saliva production because we don't need to be eating or doing anything unnecessary, so we can halt those not useful actions in order to focus on fight or flight. And then, of course, the parasympathetic does the opposite. And so we can go down this list and look at these sort of contrasting functions and how they work. And what it basically sums down to is our sympathetic nervous system focuses in on all of those actions that will help us either run away from a threat or face a threat. Those are things that are only going to be necessary while we're in a fight or a run for our lives. And then the parasympathetic nervous system gets us back to normal. So once everything is settled, once we're out of danger, we can return to our normal levels, and that's how these work together. So they work sort of in a balance to keep us right in the middle at our homeostatic point. We maintain homeostasis, which is sort of our body's happy medium point, sort of where we have not overly constricted or not overly dilated pupils. They're right in the middle. Or not too much, not too little saliva, right in the middle, that sort of thing. And we'll come back to that idea of homeostasis later on as we work through this course. The last thing that I want to mention about the peripheral nervous system is the fact that our somatic nervous system can be made up of both afferent and efferent nerves. And just to make them clear, I'm going to sort of overpronounce them and talk about afferent and efferent nerves. I'm sure people don't actually say it that way, but I find for lectures, it's a little bit easier to over accentuate so that you know what I'm saying. So we talked about the central nervous system is our brain and spinal cord. So within our peripheral nervous system, if a nerve is afferent, then it's heading towards the central nervous system. So this is usually going to be things like sensory neurons that are bringing signals from the body into the central nervous system. So that would be an afferent neuron. And then we can have efferent, means that they're moving away from the central nervous system. So that would be mostly our motor neurons. So these are just two terms that are pretty important to know when it comes to our peripheral nervous system. Before we move on to talk about the central nervous system in any kind of detail, I want to take some time to discuss how we actually study the brain. It kind of makes sense to have an understanding of how we've gathered the information about the brain before we dive into talking about what all the specific parts do and how they interact and things like that. So let's start, as per usual, with a little bit of early background history. The details of this aren't super essential, but I just want to give us a starting point so that we know where the rest of our techniques come from. So, the invention of the microscope was really important because it allowed scientists to start separating out individual neurons by hand. So they could look at what these individual cells looked like, they could look at the structures, they could kind of get a feel for their shape, and maybe a little bit of how they connected to one another. Um, Even better, once they started developing staining techniques, it allowed them to visualize individual neurons in different preserved tissues. But again, the problem here is that we're still looking at sort of dead cells that are just existing either on their own or surrounded by other dead cells. So this helped improve our understanding of structures, but when it came to function and how they integrated and worked together, we really didn't have a way of monitoring that. So, in the early 1900s, we started to see the development of reliable methods of recording electrical activity in the brain, which has sort of evolved into all of the myriad of techniques that we have nowadays that allow us to study brain functionality and what's going on internally. We can start by talking about electroencephalography, or EEG. And this is a way of monitoring electrical activity in the brain. So in this image here, you can see an EEG readout or a cleaned up readout, as well as someone who has those electrodes that are monitoring brain activity on their head. So we can see here that we have differences in activity levels based on what state we're in. So we'll talk about sort of being awake versus drowsy versus being in a deep sleep when we start talking about consciousness, I believe in chapter 5, but we'll come back to the specifics of those. But yeah, so we're basically measuring electrical activity using electrodes. And there are different kinds of EEG. You can have inter or intracranial, basically meaning that they can either be on the skull, so on the surface of the skull outside, usually just apply some kind of gel and then the electrodes on the surface of the scalp. Or you can have them within the skull. So if somebody's brain is open and you're having brain surgery or something, you can have electrodes directly applied to the surface of the brain to get a much finer resolution and get much more detailed outputs. So that can be helpful determining which parts of the brain are or are not active at different times. And one of the amazing benefits of EEG is that they have a very good time resolution because we're just measuring electrical output. So we have sort of millisecond resolution. So we can see exactly what's happening almost as soon as it happens. The problem is that when we're getting these readings, each of these lines corresponds to a single electrode that's on a person's skull. So looking at this, it doesn't necessarily tell us much about location because it's not mapped onto the brain. So we'd have to start thinking about, okay, what's the name of this electrode? Where is it located? What part of the brain is that reading? Okay, now which ones are active and which ones aren't. So it's a little bit less intuitive to look at an EEG and figure out what's going on, but it is a very good way to figure out sort of where we're seeing activity, especially if we care about how quickly we see that response. Another one that's not so much an imaging technique, but just a way of studying which parts of the brain might be involved in different processes, is to use what's called damage studies or lesion studies. And so sometimes, especially in humans, we'll see situations where areas of the brain are damaged, sometimes intentionally, in order to alleviate symptoms. So maybe if someone has epilepsy, they would have their corpus callosum severed, which we'll talk about when we talk about the central nervous system. And that can help reduce the severity of epileptic seizures. Or in the past, there were a lot of prefrontal lobotomies performed in order to disconnect the frontal lobe and the thalamus of individuals with problematic behaviors. But there are some intense side effects associated with that. And again, we'll come back to that later. But as a side effect of performing these different types of activities to help alleviate symptoms, we could then see what happened to that person's behavior after the procedure. How did adjusting this part of the brain affect their behavior before and after? We can also have intentional lesion studies. These are typically only conducted in non-human animals, but if you can damage a particular part of the brain, then that part of the brain is considered inactive. It's not working anymore. So what happens to that non-human animal's behavior when that part of the brain is no longer able to function? And more recently, we've been able to do these kinds of lesion studies in a non-permanent way. I have attended a conference with someone who basically uses supercooling. So they will cool part of the brain so that it's not active. And then when they rewarm it, that part of the brain functions perfectly normally. So they're able to perform effectively lesion studies without causing permanent damage, which is a really cool way of doing it, but also a much less invasive way of testing these things, which is always better. The next of our not quite imaging techniques is transcranial magnetic stimulation, or TMS. And if we look at the name of it, transcranial, okay, so we're going across the skull. Magnetic stimulation, so we're using a magnetic field across the skull to stimulate the brain, which is exactly what happens here. And so what we have is a very strong magnet that can be held over parts of the brain. And when those magnetic fields interact with our neurons, they can affect, either turn on or off, neurons that are held in that magnetic field. So this can allow us to either activate or deactivate, not necessarily by turning off directly, but maybe by disrupting any kind of intentional pattern within our brain. And then we can see how performance on different tasks can change when they've been affected by these kinds of pulses. So this is an example of the kind of data that you might look at. So a proportion correct. So when left to their own devices, people can answer questions pretty well, but if you deactivate part of their brain or interrupt the part of their brain that's involved in processing that information, you can see a drop off of their performance. And so you can do all sorts of neat little things where you can deactivate or disrupt parts of the brain in order to see how it changes their performance on different kinds of tasks. So that's another of those non-permanent ways to see how different parts of the brain might be involved in day-to-day tasks. Okay, so now we can get into our actual imaging techniques. So we're going to focus on some of the big ones. So you've probably heard of a CT scan or a CAT scan. This is talking about computerized tomography. So basically a computer enhanced X-ray. So we have an X-ray source, and then it's sort of the X-ray source can sort of move around the head to get lots and lots of different slices, and then those slices get put together within a computer to give us these kinds of images. So here, this is showing that there's a tumor or sort of some kind of spot on the brain that shouldn't be there, so that's been highlighted. And just as another kind of image, this is what it could look like without color. So we're building up a picture of the brain based on a whole bunch of x-rays that have been delivered. And what we care about here is we have differential absorption of those x-rays. So some parts of the brain appear darker or lighter depending on what they're made of. So this does a good job of revealing sort of the gross structures of the brain. So this gives us the big picture stuff very, very well. But you might notice that it's not giving us very fine details. So we don't have a great spatial resolution here, but it does give us a fairly good idea of the overall structures, and we could see things like tumors because they'd show up with differential absorption. So that would be fine. The next of our techniques is called positron emission tomography, or a PET scan. So this is going to use radioactively tagged chemicals to show us which parts of the brain are currently active. So usually you'll either have injected or ingest some kind of mildly radioactive chemical. So it's always short-lived radioactivity, so it's not going to be a prolonged long-term effect, and they're always something that isn't going to produce enough energy to cause problems. But what happens is those chemicals get into the blood, and then we can see how blood flows in the brain. And this is based on metabolic activity. So the parts of our brain that are more active need more oxygen, they need more resources. So there's going to be a higher blood flow. And that means that more of these radioactively tagged chemicals will build up in the parts of our brain that are currently very active. So this helps us map actual functional processes in the brain. So here, the areas that are red would be areas that are more active, and blue are less active. So we can look at what's happening in someone's brain and say, okay, what parts of the brain are most active right now? How does that relate to whatever task they're doing? Maybe you've asked someone to count to 10, or maybe you've asked someone to imagine a tree, or whatever you can do with someone who's in one of these scanners. So it gives us an idea of which parts of the brain are active based on blood flow, which is influenced by metabolism. Our next kind of imaging is actually called magnetic resonance imaging, or MRI. And we have two basic kinds of MRIs. We can have functional or structural magnetic resonance imaging. So fMRI or SMRI. Sometimes they'll just call it regular MRI if they're talking about a structural MRI. But an MRI is going to use magnetic fields and radio waves, and of course, the computer to sort of crunch together all of the outputs from the other methods. So it's going to build up a picture of our brain using those magnetic fields and seeing how they react, again, differentially with different tissues. So the magnetic fields are going to react to different densities and different types of tissues in different ways, which allows us to see the contrast between things like white matter and gray matter, or different structures within the brains, or whichever. And as you might guess, these structural MRIs are really good at detecting structures. So it allows us to get a clear, highly spatially resolved image of all of the different structures on the brain. So we can see here all of these different sort of curves and bumps and fissures. All of these different shapes are showing up really well, whereas in our earlier, say a CT scan, they're not as clearly resolved. So that was one of our problems with a CT scan, and that's definitely fixed with an MRI. And even better, if we pair a structural MRI with a functional MRI or an fMRI, we can actually get measures of activity, kind of like we just saw in our PET scan, or PET scan. So here we have sort of in the background, we're mapping on top of a structural MRI, but we're now going to specifically try and measure changes in blood oxygenation and blood flow. So same thing, more blood and more oxygen and more movement are going to happen in the parts of the brain that are most active at any particular time. So when we see these areas light up, that means that they're active because they're using more blood and more oxygen and more resources. So we can map activity and then sort of superimpose it over the structure that we got from a regular MRI. And then the last two that I wanted to talk about here are, I don't think they're mentioned in the textbook, and they're kind of weird ones, but I wanted to mention them briefly because they're cool. So the first is diffusion tensor imaging, or DTI. And this actually builds a picture of water movement in the brain. And so the idea here is it's basically like an MRI, but we're looking at not just still images. Sorry, wrong way. It's not just sort of like this where we're looking at sort of blobs of activity. What we're looking at is movement within the brain. So we're still using an MRI, but we're trying to observe blood flow because blood has water in it. And we're going to see the flow along particular neural tracts. So if you looked at one of these, actually, if you Google them, they'll actually be moving images. So you can watch if, is it going this way or is it moving this way? And it'll show you. and it gives us great spatial resolution because it's an MRI, but it also gives us that directionality of movement. So that's a really neat sort of application of an MRI. And then the last one is our near infrared spectroscopy, or NIRS. And this is again looking at changes in blood oxygenation. So blood oxygenation as a stand-in for activity level, where more activity means we're going to use up more oxygen. But they do it by shining infrared light through the skull. So this isn't done in a big machine, or it isn't always done in a big machine. It can be. And they basically look at how that infrared light bounces through the skull off the brain. And so the curvature of the light will change depending on oxygenation levels. So this is just another way to measure brain activity, but it's not as precise or as refined as the others. It's just something I wanted to mention, again, because it's kind of interesting.